So there's another DNA technology that I really want to tell you about, but before I can tell you about actually several of these technologies, we need to take a quick aside and talk about how bacteria prevent themselves from getting infected by viruses. So here I have a bacterium. And sometimes, just like us, they get attacked by viruses. The kind of virus that attacks a bacterium is referred to as a bacteriophage. Literally, this means bacteria eater, so you can tell they're quite targeted. Now, the way viruses work is that they inject their DNA into the cell that they're trying to corrupt. And their goal is that when the cell duplicates its own DNA, it will duplicate the virus DNA. And when the cellular machinery goes to use the DNA, it will pick up some of the virus DNA and it will start making proteins coded for by the virus's DNA. And that's how viruses reproduce, because viruses are just protein and DNA. Some of them are just protein and RNA. And what happens is, as the cell starts using the DNA that the virus injected, it will start making parts for more viruses, and those parts can spontaneously assemble inside the cell, and once the cell fills up with viruses, it bursts, it lyses, and then it spreads more viruses. Now, as you can imagine, this is kind of a problem for bacteria and other kinds of cells as well. And what bacteria have come up with is something called a restriction enzyme. They would like to only copy and use their own DNA, the bacteria. So they would like to restrict what kinds of DNA are allowed inside the cell. No viral DNA and only bacteria DNA. That's what they want. So what does the restriction enzyme do? It takes the virus DNA and it cuts it up like it's a chef just chopping, chop, breaks it up into these tiny little pieces. And as this is all broken up, it cannot create the full proteins associated with the virus, and that prevents the viruses from being able to replicate, because basically it's breaking down their DNA before it can be used. So these restriction enzymes, pretty useful for bacteria. But we were interested in exactly how those restriction enzymes worked, and we found something interesting. We found out that if you have a section of DNA like this, the place where the restriction enzymes will cut is very particular. It will find sections of DNA like this, which are what we call palindromes. Palindromes, if you're familiar with the term just from normal English language, are words that are spelled the same backwards as forwards. So the word race car, for instance, is a palindrome because if you spell it backwards, you still have race car. Now, obviously, this section that I highlighted, GCAATTGC, is not, it doesn't read backward here just the same, but if you read this forward, GCAATTGC, and then you read the bottom side backward, GCAATTGC, you'll see that it actually is going read the same forward and backward if you look at both strands. Remember, DNA strands are anti-parallel. So if you're reading it, say, in the 5' to 3' direction here, that means you're reading it in the 5' to 3' direction, going the opposite direction down here. So you are reading it in the correct way. Anyway, these palindromes, each restriction enzyme is made to target one specific kind of palindrome. So when they're used here, we don't just call them palindromes, we call them restriction sites. And what will happen is it will make a cut right here in the DNA molecule, and it'll make a cut right here in the DNA molecule, allowing it to cut that molecule like so. We're going to break this DNA. Now, we have a series of bases here that are hanging off the side. These can hydrogen bond, because that's how DNA is held together, is through hydrogen bonds, and they are available for hydrogen bonding. As far as the bacteria is concerned, this is where the usefulness of restriction enzymes ends, where it cuts up the viral DNA into little pieces like this. But for our purposes, we found something very clever to do with them. So for example, if I had a section of DNA from one species like this, and I used a restriction enzyme to cut it, I would get a cut that looked, something like this, right? That's similar to what we had above. And if I had a restriction enzyme from a second species, and I cut it with the same restriction enzyme, then it would find the same palindrome, and it would cut it in exactly the same way. Now I could take a section of DNA from the one species that was cut in two places with the same restriction enzyme, and I can fit it in to the DNA of the other species. Now, you remember our DNA replication, our various different enzymes that are involved in DNA replication? There was one, and it was called ligase, and its job was to ride along the lagging strand and find the Okazaki fragments and bind them together. So, using restriction enzymes and DNA ligase, we can pull a gene from any species and splice it into the genome of any other species. If you've heard about GMO crops, transgenic organisms, this is one of the ways that they are produced. We cut a gene out of one species that we're interested in, we cut up the DNA of the second species that we're interested in, we combine their DNA together like this, and then we can see if we can get a species with the right gene segment inside. There's other ways we can utilize this technology. So, if I get some more space here, we all like crime shows, I find. It seems to be a pretty universal enjoyment. If we had DNA from a suspect and DNA from the crime scene, we could take it, the DNA from both our suspect and the DNA found at the crime scene, and we could cut it up using restriction enzymes. So we could come in and make some big like this, like that. Notice that these restriction sites are going to be randomly placed throughout the genome. So one person's DNA might cut up into all these little segments like this. Another person's DNA might have more big chunks in it because the spaces between the restriction sites will be random, but they'll be unique to each person. What we can then do is load their DNA into what's called a gel, and it really has consistency of a Jell-O jiggler. It's a electrophoresis gel. That's a big word for today. Electrophoresis. It's the process of separating different segments of DNA away from each other. So what we do is we take that gel and we put a sample of DNA from the crime scene in one little well, and a sample of DNA from our suspects in different wells. Now, the same DNA samples of DNA from the same person, the restriction enzymes will be in the same spots. So the chunks of DNA it gets broken up into will be the same sizes. Different people, though, will have different locations for these restriction sites, so the size of the chunks will be different. So what we have to do is separate the DNA segments based on size. What this gel does, it has a negative electrode up here and a positive electrode down here. That creates a current like this. You might remember that DNA has an overall negative charge because of all those phosphate groups in the sugar phosphate backbone. So what happens, negative repels negative, the DNA starts moving its way through this gel because it's attracted to the positive side and it's repelled by the negative side. On the microscopic level, this gel isn't solid. It's a series of different protein strands which are crisscrossing in front of each other. It's like trying to run an obstacle course or move your way through a jungle gym or something like that. Tiny strands of DNA, the really short segments, they have an easy time getting through. So the shortest segments, will move further down the gel, and the big segments will move not so far down the gel because they're getting caught in the protein fibers of the gel. So what this does is it separates all the tiny strands of DNA down towards the bottom, and it keeps all the big strands of DNA towards the top. Since everyone's DNA is going through the exact same system, we know that. Take a look at the difference between suspect 1 and our crime scene here. Suspect 1 has this big chunk of DNA right towards the top. It's not there on suspect 1. Suspect 1 has these two relatively large sections of DNA right here. It's absent on our crime scene. And yeah, they do share some DNA in common, like this chunk seems to be in common, but almost nothing else in their DNA profile seems to match up. Meanwhile, suspect 2 has every single marker in place here. This means that either suspect 2 or their identical twin left that DNA sample at the crime scene. Now, it doesn't necessarily mean they're guilty of anything. In fact, this kind of test could just as easily be used to exonerate someone from guilt. Now, what if we have only one drop of blood at the crime scene? Is there enough DNA to run this kind of test? Maybe not at first, but we have another kind of technology called polymerase chain reaction. Polymerase chain reaction can be used to amplify a DNA sample. What does that mean? It means that it's going to copy it. Polymerase chain reaction takes the same kinds of enzymes that we use in DNA replication inside of our own cells, and it puts in basically a big machine, and it just copies the DNA and copies it and copies it. In order to catalyze this reaction, we have to do it at relatively high temperatures. So they can't use the kinds of replication enzymes that exist in our cells, in human cells. Instead, they end up borrowing some enzymes from archaea and other kinds of single-celled organisms that live in really hot water, like boiling hot springs and that kind of thing. Those thermophilic bacteria have enzymes that operate at high temperatures. They would have to have enzymes that operate at high temperatures. And those are the ones, finding those was the key to getting this technology that allows us to copy as much DNA as we want, and that allows us to run all kinds of different tests. So PCR is another good technology to know about.